Hello everyone. Currently we are discussing module 9 and in this lecture I will be discussing about viruses. They are also known as the smallest enemy. So in this lecture I will be covering general introduction about viruses followed by their unique multiplication strategies, and then also I will introduce about equilibrium and non-equilibrium viruses, and at the end the link between viral infection and cancer, because all of us we are aware that viral infection and it causes different type of diseases like flu, fever, something like that, but also viral infection can cause cancer. So let's start from the introductory part. So virus, so they are acellular infectious agent. So and they have two phases. One is non-living and the other one is living phase. Because virus, they are not cell, they are not truly an organism. Sometimes when we speak, when we talk, sometimes we mention that way, but strictly speaking, viruses are acellular. Only like they are staying, or I would say they are showing their some properties, some living properties when they are present inside the cell. So as a result of that, living phase only you will be seeing inside host cell. Otherwise, outside host cell, it's just just a non-living object. And then another important feature is virus could be considered as a simplest and hence ancient form of life. So here this life term is a problematic, because I just mentioned that whether it is living or non-living, that is the problem. Inside the host cell, it is showing some trait, and on the basis of that we can say it's living. For example, it can multiply itself, but truly it is not a living system in that way. And also it is simplest because it has not that much of complexity, only it has some nucleic acid and protein, that's all. And at this point, hence ancient I am mentioning here because it is simplest. But still, scientists believe that viruses, they are not ancient as bacteria, because I already mentioned virus need host in order to survive, in order to multiply. So as a result of that, host should be evolved even before the virus. So that's why generally researchers, scientists, they consider that bacteria, so they evolved around 3.5 billion years ago, and in compared to that, I would say virus, they came little later, around 2 to 2.5 billion years ago, because they need some host depending on so that they can multiply also. So they evolved little later, but still they are the simplest. And this is just composed of nucleic acids and proteins that already I have mentioned. And a virus is an infectious, obligate and intracellular parasite. So as a result of that, it needs to infect some host. It can be like plant, it can be animal, that doesn't matter, but it is infectious particle. And obligate intracellular parasite, without going into the host, it cannot multiply, that's why. And another important property is virus can exploit all three domains of life. So these are the three domains of life: archaea, bacteria and eukaryotes. In eukaryotes you can put, I would say, unicellular eukaryotes, and then multicellular, and here you can put plant, animals, including humans, everything. So virus can exploit all of them, virus can infect all of them, right? So although report like about viruses, their attacking archaea is much less, but virus they are depending on eukaryotic host, or they are infecting eukaryotic host, particularly human. There are huge number of examples. And then the last thing is virus, they are submicroscopic particle. So they are very, very small. As I at the beginning I mentioned the smallest enemy. So you need to have electron microscope, you need to see through electron microscope so that you can really visualize how viruses look like. So here, just for comparison, I am just showing. So this is some, I would say, some mammalian cell, okay. So this is mammalian cell. Now here, only this portion we are showing in much larger view. As you can see, these are the ribosome present in this cell, and the diameter is roughly 20 nanometer. And as you can see, smallest virus, or the virus which are in the smaller side, they are approximately 30 nanometer. For example, poliovirus. So as a result of that, a virus particle is almost, the dimension is almost like a ribosome, and you can understand that inside one eukaryotic cell, one mammalian cell, thousand ribosomes are present, and not just ribosome. There are so many cell organelles and many things. So that's why what I'm trying to explain you, that viruses are very small, but still see they are how dangerous. They are creating a lot of problem to human societies. Now, I would like to focus on some general structural features of viruses. A complete virus particle is also called virion. So this is called virion. So you will come across this term also sometime in textbook or any places. That's why particularly I am mentioning virion, although I am mostly referring virus. So now the virus is comprising of genetic material which is surrounded by a protein coat. It's very simple structure. And in some cases, an envelope is also present, but it's rare. And this envelope is derived from the host cell membrane. So when virus is, they are multiplying inside the host, and when they are coming out from the host, during that time, they acquire this envelope. So that's why I'm explaining that this is derived from the host cell membrane. And now, if you see how virus looks like, although they are very small, still look like this is the thing. In living system, you will get lot of diversity. Although I am saying that virus is not truly cell or living system, you cannot say like that, but inside the host cell, it is showing its living properties. And you will see they are so small, but they have so diverse features also in terms of their shape, size, and everything. So here, as I already mentioned, that non-enveloped virus, that means they don't have this envelope outside. For example, adenovirus, poliovirus, they are non-enveloped virus. So then what is this? So outside, this is the protein coat. This is called capsid. The protein coat of virus is called capsid. And inside the protein coat, you have like nucleic acid here. This is the simplest structure. And sometimes they might have envelope, as I already mentioned. For example, in influenza virus, in coronavirus, they have envelope outside. So as a result of that, you have the capsid, you have nucleic acid. In addition to that, they have envelope. And sometimes they have some protein which are present on the surface. This is called spike, the spike protein. Like coronavirus, influenza virus, all of them, they have spike protein. Now, whatever this simplest structure I mentioned, this protein coat can be made up of just one protein, or it could be many protein also. So that's why the variation is coming. For example, if I say TMV, so this is tobacco mosaic virus, TMV. This is plant virus, okay, plant virus. So in this virus, if you see at the center, it has one nucleic acid, and it is surrounded by protein which is capsid, but same protein, just one protein is like present in many number, and it's formed some kind of helix like structure, helical structure of this protein, and they are kind of covering this nucleic acid at the center. So some other viruses, they might have multiple protein also, and they are forming this capsid. Now here these are the some electron micrograph images. For example, this is adenovirus, and here influenza virus, as I already mentioned, they have some spike like structure. So that's why you can see here. So this is spike. And now this is bacteriophage, for example T4. As you can see, their structure is completely different. They look like completely different. They have like this is head, and this is tail, and it looks different compared to other viruses. But their basic features, they are made by only nucleic acid and protein, and some viruses they have the envelope. And now like some unique features of virus possesses DNA or RNA as their genetic material. This is some unique feature, because all other cell, I would say, living system like plant, animal, bacteria, fungus, all of them, they have DNA as their genetic material. But here I am mentioning that DNA and RNA both can be genetic material of virus, right? For example, influenza virus, they have RNA as their genetic material. Now the interesting discussion should be here, then if some virus they contain RNA as genetic material, then when this virus will multiply inside the cell, then somehow it needs to make more RNA in order to multiply. So as a result of that, you need some enzyme which can make RNA to RNA, right? RNA to RNA. So that's why they need some enzyme. It is called RNA dependent RNA polymerase. So you know that in our system during transcription I mentioned that we have RNA polymerase. This is like they use DNA as template and then they synthesize RNA. But this is RNA dependent RNA polymerase. So it can synthesize RNA out of RNA, which is only present in viruses. Okay. So the I would say the information to code this enzyme should be there in the viral genome itself, and when it will enter inside the host cell, it will make this enzyme. Similarly, some viruses they can convert their RNA into DNA. So if their genetic material is RNA, they can convert this RNA into DNA, and this is done by some enzyme called reverse transcriptase, because as you can see this is just opposite of transcription, that's why reverse transcriptase. This is again another enzyme which is only encoded by virus. So for example HIV, so they have this reverse transcriptase encoding gene. Now like this is very interesting that these enzymes, particularly this reverse transcriptase, this is very useful for genetic engineering also. Like during genetic engineering lecture I will discuss that how for routine genetic engineering approach we use this enzyme in order to make DNA out of mRNA. So we say that cDNA synthesis by reverse transcriptase enzyme. So as a result of that, those are very essential to carry out genetic engineering techniques. Now I am coming back to virus again. So they lack ribosomes or any membrane bound organelles. So they are very small. I just mentioned that their size is comparable to ribosome. So they don't have any other organelles, right? So even they don't have ribosomes. These ribosomes I am mentioning because if they don't have ribosome, that means they cannot synthesize protein, right? So ribosome is protein synthesizing machine, right? Machinery. So as a result of that, if they don't have ribosome, they cannot make their own protein. So they need to rely on host cell. And then virus hijacks host cell machineries for their own good, as just I mentioned, to make more and more virus particles. I will discuss how it is happening. Here, viral multiplication, how virus actually multiply inside the host cell. So here particularly I will give example, I will discuss this with mammalian cell or animal cell as an example. So as you can see, this is some eukaryotic cell, I can say some animal cell. As you can see, the nucleus is present here. This is nucleus. Now, what happened? The virus particle, so it first attached to the surface of the host cell. So this is the first step, attachment of virion to host cell or attachment of the virus to the host cell. Now in the next step, what will happen? The virus particle will be inside the host cell. So this is entry of viral nucleocapsid. So the process is different for different viruses. Sometimes endocytosis may happen, and that's why this virus particle go inside the host cell. Sometimes if you see bacteriophage, so in case of bacteriophage, the protein coat that is not going inside the host cell. So host cell means here in this case some bacteria, because bacteriophage, they attack bacteria. So what they do, so they just inject their genetic material inside the bacteria, and from that genetic material again new virus particle will be generated. So that the mechanism differ, but the major step here is the entry of viral nucleocapsid inside the cell. And the next step, the synthesis of viral proteins and nucleic acid. See, both are important. I just mentioned that in case of bacteriophage, the protein is not entering inside the host cell. That is true. So that I also explained in the first module, like when I was explaining about Hershey and Chase experiment, right? But the thing is, whatever the nucleic acid went inside, it has the information to make both protein and nucleic acid. So as a result of that, synthesis of viral protein and nucleic acid should be the next step. And then self-assembly of virions. So then proteins and nucleic acid, all those things should be assembled together to generate new viral particle. And the final step here is the release of progeny virions, that means progeny viruses. So as you can see, they are coming out from the cell. These are new virus particle coming out from the host cell. So this is very brief introduction about viral multiplication, different stages. But now if I discuss this in little bit in a more complex way, then it is something like this. So I will not go into much more details, but little bit more I will add here in this slide. So as you can see, this is one virus, okay, and this virus, they have the protein layer outside, this is called capsid, and also they have genome inside. As I mentioned here, genome, the viral genome is present inside the capsid. And now if you see, this virus will first attach to the receptor present on the mammalian cell surface. For example, there are many receptors present, virus specific receptor present in host cell. So, for example, I would say, like many of us, we know about HIV virus, right? HIV, human immunodeficiency virus, HIV virus. So, it recognizes some receptor which is present on some T cell. So, which receptor it recognizes? CD4 receptor. So, CD4 receptor present in T cell. So, that is why HIV virus, they attack CD4 T cell. Similarly, I would say HSV virus, herpes simplex virus, they like use different cell adhesion molecule present in our cell surface as their entry receptor. So, somehow they are hijacking our own system, our own protein, surface protein they are utilizing, and that way they are getting attached. As you can see, this is the attachment. And then whenever the virus genome is present inside the cell, then the viral genome will do many things, and this is very complex process, and it depends on what kind of viruses we are talking. So, for RNA virus, for DNA virus, and then negative strand, positive strand, there are so many complexities there. But in brief, what I want to mention here, that in one hand, from the viral genome, mRNA will be produced in order to make protein, because all the raw materials, the machine, everything present inside the host cell, including ribosome, tRNA, everything present inside the host cell. And that way, it will utilize host machinery and it will make viral proteins. So, for example, all these are viral proteins. Maybe they will form the capsid, viral proteins. And at the same time, they need to replicate their genome. So, as you can see, so this is replication of their genome. So, whenever the protein, viral proteins are ready, as well as viral genomes are already replicated, then they will assemble. This is a very complex step. Even scientists, they are not like aware about everything about the packaging part. Like this self-assembly process is very interesting for viruses. So, whatever protein and this genomic material, they will pack together, and this is a very nice self-assembly procedure, and finally you will be seeing that the complete virion here, complete virus particles, and they are coming out as progeny virus from the host cell. So, this is the overall procedure how virus multiply inside the host cell. And as you can see, I just listed their step, like first step is attachment, then entry, followed by uncoating or disassembly, so that the viral genome is available, and followed by translation of protein, and then replication of viral genome, and then packaging and assembly, followed by release of viral particles. Now, here whatever I mentioned right now, this is, I would say, most of the viruses, they infect in this way, they multiply in that way. For example, flu virus, like influenza virus, something like that. But I would say there are several different types of viruses, and their strategy is little bit different. For example, here I am trying to explain the types of viral infection and their effect on animal cells. You can consider in context to human cell, same thing. So, here whatever I mentioned, I just mentioned that viral will enter, it will multiply, and then it will lyse the host cell and progeny virus will come out. But here a little bit different way I would like to present this, that when the virus attached to the host cell surface, this is also called adsorption to host, followed by entry, and then there could be different possibilities. What I mentioned this part, that rapid multiplication inside the host cell. So, as a result of that, you will say that's cell death, and the viral particle will be released, whatever I discussed in the previous slide. And this is an example of acute viral infection. Acute viral infection. But another case can be something like this, that virus entered inside the host just like this, but the viral genome is now present inside the host cell, but host is not harmed, host cell is not dying, virus is not multiplying in that way, it is only residing there. Sometimes some virus, they even integrate their genetic material to the to the host cell DNA. So that way they are maintaining their DNA inside the host cell, but they are not immediately destroying or lysing the host cell. So this is, I would say here, latent infection. Latent infection. And some other factors might activate them, and then again from this latent infection it can go to this rapid multiplication stage, so that again it can go in that direction. And in another case, what can happen is that virus enter inside the host cell, but virus is multiplying very slowly, their process is slow, and virus, like they are getting released without cell death. So as a result of that, this leads to chronic infection, chronic viral infection. And last one here, this is although not very common, some viruses, they make some protein. So those are particularly oncovirus, they are causing some kind of cancer. So those protein, they impair the function of tumor suppressor gene. So as a result of that, those cells, those host cells, they are going into, I would say, in cancerous transformation. So I would say this is malignant transformation. So these are the different options it can happen because of viral infection. Now, viral growth versus bacterial growth. So whatever I mentioned the process of multiplication, but here I would like to focus a little bit from a different approach. So if you see here in the x-axis this is time, and in y-axis this is cell number, and whatever curve you can see now, this is for bacterial growth curve. So this is bacterial growth curve. For example, E. coli. E. coli, you know that in optimum condition, E. coli can multiply in 20 minutes. So as a result of that, E. coli, it's kind of binary fission from 2 to 4, 4 to 8, that way it will grow. And you will get some kind of this kind of curve. Initially is lag phase, then log phase, then it is going like this. But in case of viral growth, it is completely different. As you can see, so again in x-axis this is time, and in y-axis the number of infectious particle, the viral particle, I would say. So now if this is the starting point here, the infection started. Infection means the host cell is infected by some virus. Now at the very beginning, for some long time, I would say, this is called eclipse period. What is eclipse period? It is the period, or this is the lag between the initiation of infection. So here the infection happened, initiation of infection, and the point when the first virus particle is produced here. So from this place, this is burst phase. So up to that time, nothing, no viruses you can detect inside the cell or outside the cell. This is very interesting, because as you know, the way I just explained how virus they are multiplying. So virus is not like, from one virus to 2, 2 to 4, not like that, not like bacteria. So virus enter inside the cell, they are somehow like, using our own machine, the host cell machinery, to make all this raw material like capsid, protein and genome, everything. And after some time, when those things will be ready, then they will assemble and they will come out from the host cell. That is called burst or yield. So as a result of that, after some time, we will see the number of viral particle increase like something like this. It's a lot of increase there. And they are coming out from the cell and they will infect again nearby host cell. So this is the viral multiplication strategy. So as a result of that, this eclipse period, that how long it should be, that is depending on virus also. Like some virus, like they might have different eclipse phase. And also after this period, again I would say this is again eclipse phase, as you can see here. But after some time, again it can go like burst of yield, just whatever you have seen before here, I just explained. But at some point our immune system will take over, so it will most likely clear the infection. So as a result of that, no more viral growth should be there. So this is major understanding between the growth pattern of virus and bacteria. Okay, so now I will explain about equilibrium and non-equilibrium viruses. So equilibrium viruses have been associated with the host system for a very long period of time. So that means they are usually non-lethal. That means they can cause some disease, but that should be mild disease, so that host will not die. The host cell will not be completely destroyed, or I would say the host organism will not die. So that will ensure their existence. The host will help them to spread. So that is the strategy of equilibrium virus. So as a result of that, they are non-lethal, but they spreads very well. Some example of equilibrium viruses, I would say common cold virus. Like many of us, like every year we are getting common cold. Most likely sometime rhinovirus, these kind of viruses, they cause common cold. So they are example of equilibrium virus. On the other hand, non-equilibrium virus, so they have been jumped from another species. That means they are not in equilibrium with the host, and they are often lethal. So they cause different types of deadly disease even in human, and they spread poorly or they spread well. So both can happen. And I would say examples of non-equilibrium viruses, they are mostly various deadly virus. They are like non-equilibrium virus. In the next slide, I will explain with more example. So as I already mentioned, equilibrium viruses like common cold, rhinovirus, adenovirus, then measles, herpes, polio, all of them, they are equilibrium viruses. They causes disease to human, that is true, but in most cases, human, they will suffer little bit, but not that much lethal. But if you see non-equilibrium viruses like SARS coronavirus, so SARS is severe acute respiratory syndrome coronavirus, influenza virus, HIV, human immunodeficiency virus, and Ebola virus, all of them are non-equilibrium viruses. There are so many examples, I am just restricting here. Now, if you see a number of human coronavirus, right, which causes common cold with mild flu-like symptoms, they are also example of equilibrium virus. I mentioned here that SARS coronavirus, it is non-equilibrium, that is true. So coronavirus is not a specific name of a virus, it's a category of virus. On their surface, they have small spike-like structure. So those are called coronavirus, right? So now coronavirus, particularly few years back, whatever the pandemic happened to us, like that was because of SARS-CoV-2. This is again, if I write, this is a severe acute respiratory syndrome, then coronavirus, SARS-CoV-2 is a non-equilibrium virus which has jumped from another species. Most probably it has jumped from bats. But anyway, as I already mentioned, that most of the non-equilibrium viruses, they recently jumped from some other species. That's why I am just giving some example that influenza virus, their natural host is wild aquatic birds, and they are also infecting now to human. Similarly, bats, they are reservoir of coronavirus, Nipah and Ebola virus. Similarly, HIV, it came from chimpanzee to human. So now, this is the last point I want to discuss, because as I mentioned at the beginning, all of us are very much aware about virus, particularly that virus, they are the cause of infectious diseases. But here I would like to link viral infection with cancer. So some viruses which cause cancer, they are called oncovirus. Okay, the study of cancer, that is called oncology, right? So from that point, we are saying that virus which causes cancer is called oncovirus. Now, what happened? What is the mechanism here is that oncovirus, they trigger cancerous transformation of cells. How? So that viral genome, they encode some protein, right? The virus itself, they encode some protein, and that protein impair the function of tumor suppressor protein. So as you know, in our body, for example, we have many tumor suppressor genes, and they encode or they make tumor suppressor proteins. So they control our cell cycle, and whenever some some mutation, some DNA damage happen, and it cannot be repaired by normal machinery, then this tumor suppressor protein that actually detect this fault, and then those cells should undergo programmed cell death. But here what I am trying to explain, that this protein coming from some oncovirus, they impair the function of tumor suppressor protein. So as a result of that, the outcome will be bad. Let's see that one of the very good example of tumor suppressor protein is p53, and it is often referred as the guardian of the genome, and it controls cell cycle progression and initiate the programmed cell death, as I just mentioned, in response to DNA damage. So as a result of that, when p53 is inactivated by the binding of oncoviral protein, then p53 or this tumor suppressor protein cannot initiate programmed cell death, and therefore DNA damage persists inside the cell, and finally it will lead into cancer, right? So this is how viral infection also cause cancer. And I would like to give few examples also. But if you see from the viral perspective, from the point of view of the virus, this hyperproliferation of host cell and the lack of programmed cell death are beneficial, because cells, they are not dying. So as a result of that, if the cells are surviving and they are getting proliferated, like in case of cancer, then virus, they can also multiply there. So that's why I am saying that from viral perspective, this is advantageous for them. So here there are many examples. Here I am just giving some example which many of us we are aware about it. So for example, hepatitis B virus that causes hepatocellular carcinoma. So some kind of liver cancer, I would say, and particularly this Hep B, this is called Hep B virus, Hep B vaccine. So this is already in our national immunization program. So as a result of that, even in India, I would say, whenever kids they born, So they are getting administered by this Hep B vaccine, right? So this vaccine is mandatory. So that protect us from this kind of cancer transformation. And another virus is called human papilloma virus. So HPV, it causes cervical cancer. So these are just few examples of virus mediated cancer transformation, but there are more. And this is all about viruses, kind of that's done. Thank you very much. So you can follow any textbook like Prescott's Microbiology or Molecular Cell Biology by Lodis.